Okay, it's my honor to introduce Dr. Weber for this special BrainMap talk, because it's not actually a BrainMap talk, it's a spinal cord mapping talk. Dr. Weber is an instructor in the Division of Pain Medicine at Stanford University. He completed his clinical training at Helmer College of Chiropractic in 2009, obtaining a Doctor of Chiropractic. And for his research training, he got his Ph.D. From Northwestern University in 2016, working under Dr. Todd Parrish. And later he joined Stanford, working with Dr. Sean Mackey. And he also recently got a career development award from National Institute of Health. Okay, so.
Awesome, thank you. So I'm going to be talking today about my work with spinal cord functional imaging primarily, and then kind of move into some work I'm also doing in the brain. And I'm here actually for the connectivity course downstairs. And so I know there's a lot of people definitely doing some awesome things in this space. And a lot of what I'm going to talk about is doing mapping, and looking where activity is localized. But I want to move that into markers of disease and how we can actually use these maps and information to help our patients. So I'm just going to give a little bit of personal background and then talk a lot about the spinal cord functional MRI and the work I've been doing in that area. And talk about brain-based biomarkers of pain and then future directions of my research. So as already discussed, I am clinically trained as a chiropractor. Following that, I did some work at the Rehabilitation Institute of Chicago, where I was working on neuromodulation techniques as an adjuvant for physical rehabilitation of stroke patients, while I transitioned into a Ph.D. in neuroscience at Northwestern, where I worked in the Department of Radiology with Dr. Todd Parrish. Following my Ph.D., I did a 3-year postdoc at Stanford with Dr. Sean Mackey in the Systems Neuroscience and Pain Lab. And it was a really unique fit because Sean was looking for someone interested in clinical pain research and also somebody who was doing functional spinal cord imaging. And so I transitioned that quite well. What is chiropractic? It's really the evidence-informed conservative management of spinal and other musculoskeletal conditions. Main focus, though, is on neck and back pain. And our main treatment approaches are patient education, manual and other therapies, and then physical rehabilitation and exercise to treat those conditions. What really led me into research was the complexity of spinal pain syndromes. You have several different tissues that can generate nociceptive or painful information that are in the back. And any single one of these tissues can cause the pain experienced by an individual. But a lot of the times it's a combination of these, and you try to do the best you can to determine which tissues are causing pain and then applying a specific treatment to address those pain structures. But the evidence suggests that we really can't pinpoint what the actual pain generating tissues really are. And then on top of that, you also have the patient's biomechanics, their physiology, the psychology, and the social aspects of all of this coming in together that really creates that individual pain experience for that patient. And you kind of have to consider all these factors when you make a treatment plan. What further makes it more complicated is that there are several treatments that can be considered first-line treatments for back and neck pain. And usually what matters, what you first get and what you first try depends on what sort of provider you go see. So if you see a primary care physician, you're probably going to get advice to stay active. You may get ibuprofen or Tylenol. If you go see a chiropractor, you might get manual therapies and exercise and advice to stay active. If you see an acupuncturist first, you may get acupuncture. Or maybe you'll go to a center that has multiple providers and you'll get some sort of combination of multiple treatments. But any of these combinations are, any of these treatments, there's evidence for at least 1 to 2 weeks, 6 visit sort of trial to see if it helps. But you can imagine if you're a neck or back pain patient, you could, to try all of these could take, a year or 2 years to try all these different combinations if you're really not getting any satisfactory treatment. And that not only prevents somebody from seeking a treatment that's going to help them first, and it also then gets more and more expensive as they go to provider to provider. So really, it's the best we have is to try more conservative approaches first, and then moving to more invasive if that's in line with what the patient wants. But obviously, this leaves a lot of questions, open that we can't get a lot of information on. What is the best treatment for a specific patient? Will that patient improve? We don't have the information to be able to answer these questions. So I chose to go into neuroscience for my research training because it covers this big umbrella sort of other terms. So I can look at sensory processing, motor control, rehab science, pain, and clinical research. And I feel that neuroscience can provide the tools to uncover the neuroplastic changes underlying clinical pain conditions. We can help understand the mechanisms of these treatments and then have predictors possibly for recovery. And so my work is taking neuroimaging and then clinical research techniques and trying to attack pain. And as mentioned, I'm working under Sean Mackey at Stanford. Our team is really multidisciplinary. So we have a psychologist, neuroscientists, physical therapists, physiatrists, pain physicians, addiction medicine specialists, and then our physicists. And it's been a great atmosphere and lots of interesting people to work with. We also have a strong affiliation with the Pain Management Center at Stanford, which sees primarily chronic pain patients. And then we also have a strong affiliation with the Lucas Center for Imaging. All right, so I'm going to jump into spinal cord functional MRI. Who does spine imaging? Does anybody spinal cord imaging besides Rob?
Nobody does.
Okay, all right, so hopefully this will be interesting. But first I want to, so this is a lot of this, the bulk of the next few projects were the bulk of my PhD. So I just want to acknowledge Todd Parrish. He was a great mentor, and then the other team members and my PhD committee for their help on this. It was a long process. But functional MRI, we can use it as a non-invasive mapping of neural function, and it's an indirect measure of neural activity through the blood oxygenation level dependent contrast. And the great thing with fMRI is this high spatial resolution that you can get compared to other techniques like EEG. It's based on the hemodynamic response, where you have an area of increased neural activity. Your vascular system pumps more oxygenated hemoglobin to those areas, and you get a transient rise in the oxygenated hemoglobin content. And what's great for us is that oxygenated hemoglobin and deoxygenated hemoglobin have different magnetic properties, which influence the MR signal differentially. And so we can use the bold contrast to detect changes in blood oxygen level, and from there we infer that these are related to changes in neural activity. And so fMRI is an indirect measure of neural activity. With functional MRI, we're not just collecting one image, but we're collecting it over time. And so we can do some sort of time series analysis and look at those fluctuations. And there's two major flavors: task-based fMRI. In this example, we're giving somebody a thermal stimulus and trying to look at the evoked brain activity to that. And then we also have activity-based fMRI and looking at how these signals are correlated between different regions. So I'm estimating, I think 99.9% of the spinal cord fMRI literature out there is involved with the brain. But I really was interested in the spinal cord, and especially for pain processing, in that the nociceptive information comes in to the spinal cord, and we know that there's processing that already happens within the spinal cord. So those signals that get then transmitted to the brain, which then eventually become somehow, at least pain perception, and that experience, they're already modulated somewhat in the cord. And then also for motor function, which is important for many diseases to understand or try to treat, your motor neurons are located in your spinal cord. So it'd be great to have some sort of information on their function. So potential uses are for localization of sensory motor processing And hopefully we can use that as a non-invasive functional biomarker for injury or pathology, and then use it to predict recovery or know what treatment would be best for this patient. So if we, just going to go briefly through some of the anatomy of the spinal cord, but it's the tubal, whoops, sorry, there we go. It's a tubular caudal extension of the brainstem, so it's part of the central nervous system. It's encased by this vertebral column, and you have spinal cord segments with pairs of spinal nerves that then go out to your periphery to control your muscles and to transmit sensory information from the periphery to the central nervous system. And then in the cervical and lumbar region, you have these enlargements, largely due to the increased number of neurons for your extremities. And then there's also this left-right symmetry. I'm going to dive a little bit more into the anatomy or neuroanatomy, but I'm going to keep it really general. We're just going to break the spinal cord into quadrants from the point of this talk. So usually I'm breaking into the dorsal horn and the ventral horn. The dorsal horn has more of a sensory sort of role, and we're going to discuss, and that's where the afferents come in and make their first synapses. And then also the ventral part is where the motor neurons are housed, which then go out and create muscle contractions. And it's surrounded by, that's the organization of the brain matter, but it's surrounded by ascending and descending white matter tracts that carry that information, superior and inferiorly. So I started my PhD, there's about 120 studies. Now there might be 150 studies. And in spinal cord fMRI, and it sort of spans everything. So there's human, there's animal studies. They've looked at a ton of different sensory stimuli, motor tasks, resting state. There were a couple different techniques. Some people are doing classic gradient echo EPI, bold imaging. There's other camps that are doing spin echo imaging, proton density imaging. They've been some studies in healthy and pathological states. And then beyond just sensory and motor tasks, people started to look at higher level cognitive things such as emotion, attention, placebo, and nocebo. But I would say that a few of these studies have actually provided any physiologically relevant information at the time. And I would say, I think some of the issues with bold and the statistical analysis of bold imaging, you can find innovations like in the dead salmon, for example, depending on how you analyze it. And one of the issues with it is that no one had anatomically shown any specificity of the activity or tried to quantify where the location was and is it in an area that we actually are expecting it. So it kind of leaves me to wonder, are these false positive activations? Are we really seeing a signal, or is this just noise? And then the other issue is that reliability had not been assessed either. So my question is, is this really, can we detect a strong signal in the spinal cord, or is it really just noise? And so I wanted to demonstrate that there's anatomical specificity of the signal, that the activation rate is greater than the false positive rate, there's reliability, and that the signal would be proportional to some sort of stimulus or task intensity, which has been shown in the brain. And to do so, I did this with simple motor and sensory tasks. So the reason why I feel that spinal cord fMRI is maybe a decade behind brain imaging or more, but is there's several technical challenges, and it's really hard to image the spinal cord. One is that the small dimensions of the spinal cord, where we're looking at a centimeter in diameter, makes it challenging to image. We can image high resolution, but then we're also getting a signal to noise ratio hit. And then there's also magnetic field inhomogeneities due to changes in bone tissue interfaces, which can lead to an inhomogeneous magnetic field, and then you can get artifacts with classic gradient echo EPI imaging, such as some artifacts like that, and get signal drop dropout and distortions. And then the other big issue is noise due to the cardiac, respiratory, cerebral spinal fluids. Swallowing will induce some noise, and then just motion from the patient. So this is a great inequity PI sequence, but you can just see all this sources of noise here, and just it's a very challenging area to image. And then on top of that, there's also analytical issues in that SPM and FSL, all the software packages tools are designed for brain imaging. And so there's challenges with things like motion correction and also spatial normalization, among every single other stuff. So I spent a couple of years trying to work on imaging techniques, doing all sorts of things. I looked at proton density imaging, did some spin echo imaging. I even did a T1 weighted study and injected Ablavar, and trying to see, could these be sort of better contrast mechanisms than some of the issues we were having with the grade inequity PI. But what we found that worked best for us was to use this reduced field of view grade inequity PI sequence that was available on the new Siemens Prisma scanner. And so the advantage of this sequence uses a spatially selective excitation pulse. So you can reduce the number of phase encoding steps, so you can one, image faster, and also and reduce some of the distortion with higher resolution imaging. But we could cover essentially the entire cervical enlargement with 1 by 1 by 3 millimeter voxels in a TR of 2.5 seconds. We also used a sat pad cervical collar to try to increase the homogeneity of the magnetic field across the neck. And so here's an example of the setup. And here's an example of some motion corrected data. You can see, you can point out see the spinal cord pretty well. There's good CSF to cord contrast, so you can identify it. That can be a challenge. And if somebody has some sort of degeneration in the neck, that contrast between the cord and the other structures can be difficult to see. And then here's an example of the mean motion corrected image. And if you hallucinate very slightly, you can start to make out the boundaries between the white matter and the actual gray matter, which I think is pretty cool. You can see it more obvious on the coronal section where you see these bands here. But it looks pretty good. And then here's a TSNR image. I think usually motion corrected TSNR ranges between 15 to 25 with the process of the way I'm doing motion correction in this imaging. And then the other biggest issue is physiological noise. What we've done is used the retroact, so we collect the cardiac and collect the pulse data and use that to model what we think the signal fluctuations are going to be in the image due to that. There's a paper by Jeff and Brooks, and this was done specifically in the cord. And so they recommended, based off that paper, to use this 32 regressors. And then also I include in a CSF regressor too into the this noise matrix, design matrix. And that is manually done in. So I have to go manually in and draw this. On all these images, I have to manually go in and identify the cord and then manually create a CSF mask because there's not really a way to automatically do it. And then another challenge is motion correction. I think the motion correction techniques seem to work pretty well with brain imaging for sure, except if you get some artifact from excessive motion, trying to figure out how to get around. That's still an area of research. But the challenge with the spinal cord is that we're no longer working with a rigid structure. And you have this slice-dependent motion. So for example, here, we'll look at volume 72. You can kind of see where these different slices here at volume at this time point, but then you look at this time point and the displacements are a lot larger. And a lot of that's due possibly to the respiratory cycle. So as you breathe, your chest is moving up and down, and you start to get non-rigid motions within the cord. So what I've been doing is I do a sort of like a three-phase, I guess I have it here listed as a three-phase realignment, but I'll go in and do a 3D sort of volume correction. And then I will split the image into slices and then do a slice-wise correction for every single slice. And I also use, if you remember from that video before, there's a lot of non-rigid deformations that happen outside the cord. So I use a mask to weight the reference image so that the motion correction is confined just to that region. And I'll also say just now is that all these pre-processing techniques really haven't been looked into as far as which ones are the best. So we don't know which ones are optimized. So there's probably better ways to do this, and it would be that should be an active area of research. For me, one of the issues is just getting a large enough data set to start analyzing it and trying to figure out what are the optimum techniques. And then the other issue is spatial smoothing. Now, this isn't functional imaging. This is just a high resolution T2. But to get the point across, here's no smoothing. And the issue is if you start to use you do too much smoothing, and it's a small structure, so you'll start to smooth in the surrounding CSF. Since you have this, it's a cylinder, you can try to do the smoothing so that it's just along the cord, or more superior inferior, which is shown in this example. But here, if you go this is only along the z-axis smoothing, and you get nice smoothing up here, but where you get down into this curvature part down here, then you start to, bring in CSF into that signal. But there are some new techniques now available where you can just smooth along the cord, and usually using an isotropic smoothing kernel. I've gotten around this in that I do my denoising now, motion correction, denoising, and then I warp everything to this template, which I'll show you on the next slide. And then I apply smoothing once it's in template space. And so just in the last, 2014, a whole new set of tools became available specifically for spinal cord imaging. Before that, people were working using SPM or FNIRT and FSL to try to do normalization to a template that usually was probably produced within the lab. But we now have, there now exists a spinal cord template. This right here is an older version. There's now this one called the PAM50 template, and it has the cervical all the way down through the sacral segments of the cord. And so just like in brain imaging, we have our templates, so now we can do spatial normalization. So we register the template to the cord, and then we can co-register any sort of other imaging modality to that, usually a high resolution anatomical, and then we can warp it to subject space. And this is all due to the spinal cord toolbox, which is developed by Julian Coleman Todd's group. And now we even have, we have probabilistic white matter, gray matter maps. There's information about spinal cord level, and there's also a white matter and gray matter atlas that you can use to extract information from specific regions of interest. All right, so I'm going to talk about work from this project, which was done with Todd Parrish at Northwestern. And so this is a motor study, and the neuroanatomy, the motor system is at the lateral cortical spinal tract, which goes from the primary motor cortex, comes down, decussates in the medulla, comes back through, and then synapses onto your motor neurons. And this is the major tract involved in voluntary control of the limb movements, and it's highly specialized in primates, and it makes monosynaptic connections to motor neurons. And yeah, and then your motor neurons are located on the ipsilateral side of your spinal cord. So the motor neurons for the right hand are located in the right ventral horn of the spinal cord. And so what we want to do is a pretty simple study where we have 11 healthy subjects, and they're going to perform a motor task where we immobilize their right and left wrists with a plastic brace. And then they did alternating block design task of right wrist flexion versus left wrist flexion. It was really, really simple. We didn't know how much data we needed to collect, so we collected a lot. So we got 30 minutes of data, so 6 5-minute runs. We'll go through some of the results. The full results are in the full paper, but we expected that we would see activity located to the ipsilateral, anterior, posterior spinal cord. So if they're moving their right hand, we should see right-sided activity. And if they move their left hand, we should see left-hand activity. And that's what we saw, which was great. I knew that I was going to graduate, at least sometime in the near future. But this took us, you know, I can't stress it, took at least 2 years before we got to this point. But the red-yellow blobs are left or left greater than right, and then the blue, light blue blobs are right greater than left. And this is where you can see, so here's a coronal section of the cervical spinal cord, followed by two laterals, either in the right side or the left side. And with left-sided wrist flexion, we're seeing left-sided spinal cord activity. With right-sided wrist flexion, we're seeing right-sided spinal cord activity. The superior inferior distribution of that activity is larger if you contrast left versus right, right versus left. And if we increase the statistical threshold, you start to see that the signal appears to be higher in the ventral part of the cord than the dorsal part of the cord. And I also just want to say that this is with a 4 millimeter smoothing kernel, because I hadn't yet figured out how to get FSL to do an anisotropic smoothing kernel at that time. So probably a little over smooth than what I'd want to do. And then this is just graphically showing that you have this laterality between the tasks and the location within the cord. And not only can we detect this laterality at the group level, if we went in and then extracted, you know, the location of the activated voxels within the cord at the subject level, you could start to see evidence of, you know, the left-sided activity. The subject level was more on the left side, the right side was more on the right side. And this also shows some results from, I did some control analyses in here that I'm not going to go into, but you didn't see any sort of lateralization of the signal with these control analyses, but you do when you see the task. It's pretty strongly lateralized. Next, we wanted to look at reliability. And so we had data from 6 runs. We had analyzed those all separately and looked at the group activity. Again, the spinal cord's very noisy structure, so it's a hard signal to detect. You can see how variable the blobs are across these different 6 runs. But there does seem to be evidence for this laterality of the signal for each of the runs. And these are, for this analysis here, there's no correction for multiple comparisons. And this was this task free control analysis I didn't show any laterality of the signal. Next, we want to, you know, get a little, maybe try to find some more evidence that there is a signal there. There's some meaning in these activation maps. So we did a multi-voxel pattern analysis to try to decode whether it's the left or right task that was being performed. So what we found is that it was above chance, not very much above chance, but wouldn't be diagnostic, but there was enough information to, it's like around 60% here. It'll get a little bit, I'll show a little bit better results in the next slide, but at least that you're able to decode what task is going on better than chance based off of these activation maps at the subject level. Just another way to kind of confirm that there's information that we're detecting. Since then, I've done some work with spatial smoothing and applying anisotropic smoothing kernels along the cord. And so this is the same data set, but now this is with a 2x2 by 6 millimeter smoothing being performed. And the activity is much more localized to the gray matter than before. And so this is for the left motor task, and now this is for the right motor task over here. And if I do that same multi-voxel pattern analysis with, this is leave one subject out cross-validation, there is about a 70% accuracy of classification between the left or right tasks. Okay. So the other main project was to now look at thermal stimulation. And one of the nice things with thermal stimulation is that we can then modulate the intensity of the stimuli and try to get that relationship between signal intensity and the bold signal or the activity. The neuroanatomy for this task is the spinal thalamic tract, also known as the anterolateral system, which is carries information about temperature, nociception, and crude touch, where you have your peripheral afferents come in from your afferents from the periphery come in, and they synapse in the dorsal spinal thalamic neurons and in the dorsal horn, which then, sorry about that, but they decussate immediately, and then they go up and travel up to the thalamus, and then go to somatosensory cortex. It's more complicated than that. There's interneuronal connections within the cord. These project into brainstem areas too. They go to multiple areas of the cortex, not just somatosensory cortex. And then not shown here is also you have descending controls that also modulate these signals, either descending inhibition or facilitation. But the idea would be that the primary synapse is in the ipsilateral dorsal horn. So that's kind of where we'd be expecting to see the activity. So we had 12 healthy subjects where we did right ventral lateral forearm stimulation, which is about the C6 dermatome. And these participants received either warm stimuli at 43 degrees centigrade, or they received painful stimuli that was a temperature that provided 65 out of 100 pain rating. And again, we weren't sure necessarily how well to power this, so we collected a bunch of data. So I think we have 6 minute and 40-second runs. So almost 42 minutes, I think, worth of data, something around there. Did sort of the same analysis I did with the last paper. And then we hypothesized that the activity would be localized to the dorsal ipsilateral spinal cord, and we'd expect the signal change to be proportional to the stimulus intensity. So here's group level activation maps. It's kind of set up the same way. We have 2 coronal sections and 2 sagittals for the painful and warm stimuli. Again, is a right-sided stimulus, and right side left, left side right. You can see, look at the painful, you can see this really lateralized column of activity on the right side. But to our surprise, there's also quite a bit of contralateral activity that we're seeing with the task. The warm, I think we really are the warm stimulus, we're kind of almost at that threshold of, I don't know, just detecting something. These are corrected for multiple comparisons. I'm going to show the uncorrected. Just because there is this pretty big cluster of activity on the right side for the warm stimulus also, but it didn't meet the correction. It's also something that hasn't been explored very well in how to do multiple comparison corrections in the spinal cord. So there's evidence for this right-sided activity, but we do see a lot of contralateral activity. We also see a big ventral blob up higher in the cord, and it's interesting that it's how do you do a purely sensory task when you still have possible reflexive motor activity also. This thing was pretty intense, and we did a lot of these, so it was, I could see where you may get some reflexive activation in the shoulder area, back of the arm to try to move away from the stimulus. That might be some of it. If we look at where the activity is localized, and we see that there does seem to be more activity located dorsally in the cord than ventrally, but we didn't see a strong laterality to the signal. And then this is looking at the subject level, and we see a very similar pattern where we don't see laterality, but we do see more dorsal located activation. Next, I wanted to look at how the activity is different between the warm and painful stimulus. And so we see that there's a higher number of activated voxels due from the painful stimulus, and also a higher percent signal change due to the painful stimulus compared to the warm. Some of this may not necessarily be just to the change in intensity of the stimulus, but could be due to the now a reflexive withdrawal also. Weren't able to tease that out. This is looking at contrasting the group at level activation map. So this is painful greater than warm, and you can see where there's activation for that contrast. If we reverse it, we don't see any significant activity. And then this is doing a leave one subject out classification task. And we're getting accuracy above 65% between the warm and painful tasks. So it's just kind of giving more evidence that there's information in there that can tell us differences between these two stimuli. And then we looked at reliability across these runs, similar to before. And you'll see that there appears to be even more variability in this thermal stimulus. And it could be that the sensory stimuli, there's just a lower signal change compared to the motor task. But this is for the warm stimulus. You do see that there tends to be more dorsal activity than ventral activations. This is looking at how the number of voxels and the average Z-score of those voxels is changing across the runs. Nothing interesting here with the warm stimulus, but if we now look at the painful stimulus, if the big activation maps look similar, it's pretty variable. But you do see this trend towards a higher number of activated voxels as you go along with the experiment, and a higher average Z-score of those voxels. And we didn't record this. In hindsight, we should have. But anecdotally, it was getting more painful, and likely due to sensitization from the repeated stimuli. This is just looking at the center of gravity of the activation maps across each of the trials for all the subjects, where each dot here is for a specific run, and then the black X is the average across all those runs. I think it's from the average activation map across all of those runs. But you can just see that there's quite a bit of variability across the runs. You do see at the dorsal, looking at ventral dorsal, the center of gravity visually appears to be more dorsally located than ventrally located. So if I kind of look across these two studies, there appears to be some anatomical specificity, definitely from the motor task. The activation rate appears to be greater than the false positive rate. I didn't really talk about some of the control analyses we did, but they also support that. Reliability, I see that laterality appears to be there with the motor task. Sensory task, doesn't look that reliable. And then the signal being proportional to the stimulus or task intensity. We seem to show some evidence of that in the stimulation study. So we're pretty pleased with that. So the next project I'm going to talk about was inspired by Robert Berry's work, which this was, I think, my third year of my PhD. This paper came out. That identified these bilateral ventral horn networks and bilateral dorsal horn networks within the cord at rest. And this has now been replicated. Rob's replicated it a couple times. I know Oxford Group replicated it also, and then we have evidence supporting that it exists also. But we're doing resting state functional activity within the cord. I created this. We're using Pearson correlation here. My method for this was I created this sort of lattice of ROIs that spanned the cervical enlargement, and then extracted the mean time series, and then created correlation maps across those regions. And here on the left is across all those regions and their correlation between those. And then this is now averaged across all the superior and inferior ROIs, kind of make a little bit more sense of it. Again, what Rob showed was that you have this bilateral ventral network and this bilateral dorsal network. And down here, we're showing evidence of that, where we're seeing evidence of this ventral and dorsal also within our group of 15 healthy controls. We showed some evidence of a ventral dorsal within network, so that would be within left ventral horn, left dorsal horn, and the opposite. Not sure if that's due to smoothing that was applied, or actually, I didn't actually smooth this data, so no, not with the AI analysis. But it could be possibly from the normalization that we do. So the normalization straightens out the cord, puts it onto that template, but there is a lot of interpolation along the anterior posterior sort of, yeah. I don't know, as it stretches, I believe there could be some smoothing along the anterior posterior axis during that process. And that might be why we're seeing evidence of this here. Here's some connectivity maps across these different ROIs and showing evidence of this dorsal and ventral connectivity. We also looked at the extent along the superior inferior axis, and maybe to our surprise, I guess we didn't really know what we'd find, but it seems like the activity is pretty localized to just around that ROI. So it's not, the C5 right dorsal horn doesn't seem to be connected to the C7 dorsal horn. I don't know if, Rob, have you looked at that?
Yeah, I looked at it a little bit. I never published it, but it really does seem to be specific to the particular level. There seems to be a lot of crosstalk across the side and down different levels. It's fairly localized.
Yeah, and you know Nawal Kanani? Yeah. She also looked at this and found similar, very localized along the superior inferior axis, which is interesting.
Ken Kwong and Paul also showed that. I think everyone expected there to be correlations along the cord, and thus far, as far as I know, no one's found it. So it's a little bit of an open mystery of what's going on.
Yeah, it's interesting. So then I also had matching thermal stimulation data sets from the thermal stimulation study. And we looked at how the connectivity changed with that right-sided thermal stimulus. And what we identified is that there was a decrease in this dorsal connectivity, and also showed evidence of increased global efficiency and decreased modularity as you do with thermal stimulus happening. And I guess the idea is that you have a unilateral thermal stimulus, and maybe it's throwing off some of that connectivity. I did also look at changes in connectivity maps, but I didn't see any differences in connectivity maps. It's also a pretty small group. So we are, I think I'm doing, what time? Up till 10, is that okay? Or 5 more minutes? Yeah, we'll get through this. We're now doing some work with simultaneous spinal cord-brain fMRI. This was led by Hassan Islam. This is his Ph.D. project. Christine Law has done an amazing job. She's moving this project on, and we recently did R1 to look at use this in fibromyalgia patients, but also look at pain signaling, and should be some interesting things we can find in comparing it between the brain and spinal cord. But the big major issue is shimming and trying to shim both of these structures at the same time. And I can't necessarily go into the physics in this, but they calculate a field map, and from there they can calculate the specific shim that's needed for each slice. And here's an example of using this with healthy controls, and they're doing a bilateral fist clenching task. And brain activation pattern looks like your classic motor activation, but then we're also seeing really strong ventral horn activation across these subjects. There's also a lot of stuff outside the cord too, but we didn't. There could be some motor or muscle activation too that's linked with when you're really clenching your fist. So I'm just going to finish up here, just talking about my work in pain, but chronic pain is a big burden on the healthcare system and families. And it's intertwined with this opioid crisis, and a lot of people who get addicted or misuse opiates start to take them for pain conditions. And neuroimaging has, in the last 2 decades, has really expanded our understanding of pain processing in the brain. It's made it much more complicated than I think we thought 2 decades ago. And there's many regions involved in different aspects of this, well, specifically involved in different aspects of the overall pain experience. The goals of neuroimaging pain research are really to try to get the mechanisms, try to better diagnose what's going on, and then have some sort of prognostic measures. I hope we can use this somehow to help how we treat spinal pain. But then also, if we find the brain regions that underlie these questions, we may be able to target them with neuromodulatory techniques or pharmacological treatments. So just going to touch last project I'm going to go through. I don't know, some of you might be familiar with this. This is a neurologic pain signature. It was published by Tor Wager in 2013. But it's a multivariate brain-based model that's been shown to be sensitive and specific to physical pain. It was trained on thermal stimuli in healthy controls. But if you use this model, it predicts what pain that person's experiencing. It's been validated in quite a few other studies also. And so Jim Elliott put me in touch with Cheryl Sparks. So they're both physical therapists, researchers, and they had a data set where they did mechanical stimulation to the painful mechanical stimulation to the index finger, and did that pre and post thoracic spinal manipulation treatment, and had these activation maps. And we had a meeting where, like, how could we repurpose this data set? And I was reading up on the neurological pain signature and was, I thought, hey, let's try to see it does this signature change pre and post thoracic manipulation. And so here's just the example of the spinal manipulation technique. They were able to perform it in the scanner without even removing the patient from the head quo. But there's 2 studies here. One, yeah.
So you're performing like manipulation in the scanner without moving the patient. Can you go into that a bit more?
Oh, I wasn't there for it.
Oh, I was like, wow, that's amazing.
But yeah, they had like a space below with a foam pad that they could pull out, I believe, and then were able to maneuver them. And it's done supine, like that's in this picture. So, and so we had 2 studies here. One was in healthy controls, and one was a small randomized controlled trial. I'm just going to go through this a little quick. But what we can do is take the first level activation maps to the painful stimulus, do the dot product or weighted average with the neurological pain signature, and we get a marker of activation. In the first study, there's pretty clear evidence that there is a decrease in evoked pain and a decrease in NPS response shown here, and it correlates with the change in pain across the subjects. But then if we look in the neck pain group, it gets a little muddled and not as clear. You can see there's no change in evoked pain. Neck pain does seem to decrease, and NPS activation. So maybe there's something going on there, but if we look at correlation between these, it's weak and there's really not a pattern. So this just summarizes that. But it leads me to think of is how can we better image clinical pain? And it's challenging. There's been 3 things out there in the literature. One is you have people track their neck pain, let's say, while they lie in the scanner. And then you can use that tracking to try to regress out what areas are fluctuating according to their clinical pain. There's issues there, though, as you're having somebody track it. So how do you get rid of that motor response also? There's also ways to evoke clinical pain. One is you could do ones with irritable bowel syndrome, and they'll put a bladder in the rectum and then expand it. And you can expand it and modulate the pain experience while they're in the scanner. Other ways are to do maybe like an exercise procedure to increase pain, or maybe not exercise, or some sort of straight leg raise or something like that. Another is resting state functional connectivity. This is out of here with Vitaly's group, where they did a exercise-inducing sort of procedure, and then calculated resting state functional connectivity with this heightened clinical pain experience. And we're able to show really well that they could classify between these individuals and predict their pain level using a combination of resting state measures and also some physiological measures too. I've been working on this. I've put together a data set of about 500 subjects, maybe almost split 250 healthy controls, 250 chronic low back pains, and trying to build a classifier that can predict between whether it's somebody with health control or chronic low back pain. Work in progress, but if you can build a model, then maybe I could use that as look at how that model changes with treatment. It's sort of the idea, similar to the neurological pain signature, but something really focused on the clinical pain. And then this is just a quick plug for a project I'm doing, where we're bringing in neck pain patients with cervical radiculopathy, which means that they have some sort of pathology with their nerve root that's causing sensory issues and weakness in their arms. And we're going to track these individuals longitudinally over a year. I'm going to do baseline imaging and then do a repeat at 12 years later with brain imaging, and I'm pretty happy with the quality of the spinal cord imaging that we're doing also. So it'll be structural and diffusion and functional resting state. That's all I'll go, and I don't want to go over, but let's do those last two slides just to give them some credit.